Hi, everyone. My name is Mary Maranga from the Malopolska Center of Biotechnology. And today I'll be giving a talk of our current research on the function annotation of metagenomes and microbiogenomes. This is a collaboration between Malopolska Center of Biotechnology, Broad Institute, and the Flatiron Institute in New York. I'll start by giving a brief introduction of why protein function matters. As we know, the human gut microbiome plays an important role in health and metabolism. And so far, the function potential encoded in the gut microbiome remain poorly understood. And this is mainly because only 50% of protein coding genes may be functional annotated. This is the functional information coming from experimental sources and computational inferences. And this low function annotation coverage poses a major challenge in linking specific genes to disease phenotypes. Our main goal for this research is to function characterization of human gut microbiome. So I'll be talking about how we are annotating metagenome assembled genes to help us understand protein functions better. For the function annotation, we are using deep learning based method DeepFRI, that is the deep function residue identification. DeepFRI uses graph convolution networks to annotate proteins. It takes input protein sequence and structure and outputs the gene ontology probability. This method was developed by a collaborator from the Flatiron Institute, and it has been shown to annotate proteins at higher coverage and accuracy in comparison to methods like BLAST and DeepGeo. I'll now briefly take you through our pipeline. Our workflow integrates de novo genome reconstruction, taxonomic profiling, and functional annotation. Here we perform analysis in different phases. First, it takes input short read data in terms of FASTQ format. We then perform metagenome assembly using MegaHit to generate contigs and beam these contigs into metagenome assembled genomes using MetaBat2. We then do quality control using CheckM. We further perform taxonomic profiling using the genome taxonomy database. For the functional annotation, we first predict genes using Prodigal, and then cluster these genes based on sequence similarity at 95% using CD-HIT to generate our non-redundant gene catalog. For the functional annotation, we use two complementary approaches. First approach is the deep learning method DeepFRI. Here we use CNN-based DeepFRI. We then benchmark DeepFRI using a commonly used orthology method, eggnog. We further quantify our gene abundances by mapping short reads into our non-redundant gene catalog using KMA. This is what we use to quantify our gene functions. We further have developed a custom Python script that perform mapping between our non-redundant gene catalogs together with the function annotations and the taxonomy information coming from the GTDB. So like I mentioned earlier, our gene catalog is 95% non-redundant. So we propagate gene functions within each cluster and annotate genes within marks. And this helps us to reconstruct the functional potential of individual species. We tested the feasibility and reproducibility of our workflow using the immune microbiome cohort. This data was collected by Tommy Vatanen. This is the infant gut microbiome in type 1 diabetes collected in Finland, Estonia, and Russia. And looking into the assembly and gene prediction statistics, we assembled approximately 17 million contigs from the immune data. And we reconstructed around 7,000 metagenome assembled genomes, of which 2,000 were near complete genomes, meaning that they had a completeness of greater than 90% and a contamination of less than 5%. Our non-redundant gene catalog consists of 1.9 million genes. So like I mentioned through our workflow, that we integrate gene homology from our gene catalog and the taxonomic information coming from GTDB. So we further try to provide insights into the following questions. Are non-95% gene families taxa-specific? Here we stratify these gene families into various levels, species, genera, and family. And we found a notable portion of gene families that has multiple taxonomies assigned, 3% at family level, 11% at genus level, and 14% at species level. Here we hypothesize that this may be due to mechanisms like horizontal gene transfer, where we see species exchange genes within themselves. And there was also a notable portion of genes that could not be assigned to any taxonomy. The first step of our functional notation is validation of DeepFRI and EGNOG predictions. Here, we only consider the molecular function branch of gene ontology, and we take DeepFRI predictions above 0.2 threshold to be significant. So the first step of our validation was to compare the concordancy between both methods using Shannon information content. Basically, Shannon information content measures the specificity of our geoterms in the context of the entire annotations. And it is expressed as a negative log probability of a geoterm. So meaning that terms that are annotating many genes tend to have lower information contents, while infrequent occurring terms have high information contents. So looking on the graph on my right, the gene ontology is organized in a hierarchical way, such that if a protein has a label mRNA 5' UTR binding, it automatically inherits all the parent terms that are associated with it. And as we go up the tree, we get more general terms with a lower information content. But when we go down the tree, we now get more specific geoterms that have a higher information content. So this plot here shows difference in information content between DeepFRI and EGNOG. On the y-axis, we have the genes that are annotated by both methods. And on the x-axis, we have the information content levels. And the blue bar here shows DeepFRI annotations, and the brown bars show EGNOG annotations. From the trend, we see that as we go to high information levels, we see a gradual decrease in the number of genes annotated by DeepFRI, while EGNOG tends to give more specific geoterms. So DeepFRI here increases the annotation coverage. So it's able to annotate more genes despite having a lower information content, or they are rather less specific compared to EGNOG predictions. Our next step was to compare the concordancy between DeepFRI and EGNOG. Here, we stratify annotations into 4 categories. The first category is concordant. Concordant, what we mean by concordant is we look at genes annotated by DeepFRI and EGNOG. We see whether genes obtained functions at the same information content level. By discordant, it means the annotation disagrees, but annotation exists for both methods. We also have DeepFRI unique and EGNOG unique, is where gene only obtains information from one method. The plot here shows the proportion of concordance between both methods. That is the concordant annotations and discordant annotations. And from we see very high concordancy between EGNOG and DeepFRI, on average 70%, meaning that DeepFRI is able to give reproducible gene ontology terms that can be used to understand the biological problems. The plot on my right shows the concordancy as well as the unique annotations. So we see that DeepFRI, it's able to give more unique functions, meaning that it's able to increase the annotation coverage in comparison to EGNOG. And now looking into the per gene perspective, we see that in total, nearly 99% of our gene catalog obtained functional information from DeepFRI, while 12% obtained similar annotations from EGNOG gene ontology. Further, we wanted to complement these functional annotations by considering other functional information coming from the EGNOG free text. And for EGNOG, it gives other functions categories other than gene ontology. So it provides methodologies and as well as EGNOG free text. This is the functional information consolidated from PFAM and smart databases. And with eggnog free text description, we see an increase in annotation coverage by 72%. Now looking into the comparison of the gene sets annotated by DeepFRI and eggnog, this Venn diagram shows these comparisons of genes annotated by DeepFRI, eggnog gene ontology, and eggnog free text. We see approximately 1.3 million genes that are shared between eggnog free text and DeepFRI, and around 219 genes, 219,000 genes that are shared between DeepFRI and eggnog gene ontology. So we see by using both methods, we're able to increase the annotation coverage of the metagenome assembled genes. The gene ontology is hierarchical, meaning that a term can have multiple associations to a broader parent term and a more specific parent term. So we further filter out our gene ontology terms using a subset of informative geoterms. This informative geoterms is obtained by traversing the geotree from root and selecting terms that satisfy a certain criteria. That is, our terms are associated with more than 2,000 UniRef protein families, and its descendant term contains less than 2,000 UniRef protein families. By further filtering these geoterms, we reduce the number of genes that are annotated by both methods. This Venn diagram shows a comparison of genes that are annotated by eggnog and DeepFRI using only informative geoterms. So we see around 84,000 genes that are common between both methods and 281,000 genes that are unique to DeepFRI and 72,000 genes that are unique to eggnog. So we now wanted to evaluate the proportion of gene abundance, metagenome gene abundance with functional annotation by both methods. First, we calculated the gene abundance by mapping short reads to our non-reader gene catalog using KMA and normalizing these relative abundances in copies per million. With DeepFRI, we see an increase in annotation coverage at around, on average, 25%, while eggnog gives annotation coverage of around 14%. So we see DeepFRI is able to increase the annotation coverage of metagenome assembled genes. And now looking into how DeepFRI and eggnog performs across taxonomy, this plot shows phylogenetic placement of high quality metagenome assembled genomes. And on the inner ring, these are bacterial classes, various bacterial class. The second ring shows genes annotated by DeepFRI, and the third ring shows genes that are annotated by eggnog. So the outer bar here depicts gene families for each bacterial class. And from the pattern, we see that eggnog is able to annotate genes coming from well-studied organisms such as E. coli, while DeepFRI is less sensitive to taxa. Now looking into the per genome perspective, we reconstruct one genome in a reference-free manner using the metagenome assembled genomes. So we recruit near complete genomes, that genomes that has greater than 90% completeness and less than 5% contamination. We consider only species that contains more than 10 genomes and define code genes that are genes present in greater than 90% of our marks, and accessory genes that are genes that are present in less than 90% of our marks. Further, wanted to evaluate the functional repertoire encoded in the core and accessory genome. This bar plot shows size of accessory and code genomes stratified by eggnog and DeepFRI annotations. That is the known versus unknown functions. The blue bar shows co-annotated. The orange bar shows co-unannotated. The green bar shows accessory annotated, and the red bar shows accessory unannotated. Eggnog is shaded in gray. So we see the same pattern as the phylogenetic analysis, where eggnog is able to annotate more functions coming from well-studied model organisms such as E. coli, while DeepFRI tends to annotate more genes coming from Bacteroides genera. And our results actually could be used to guide further analysis aimed at uncovering potential mechanisms for diversity in these Bacteroides genera. So to wrap up, I've shown you how we are using the deep learning methods to annotate metagenome assembled genes. And further, we are now integrating structure-assisted predictions to help us improve the accuracy of our predictions. And our workflow is now available for public use on GitHub. I would like to thank all our collaborators who have made this work possible, especially our collaborators from the Broad Institute and the Flatiron Institute. I would like to thank you all for listening.